and biochemical mechanism of abiotic stress tolerance in plants, including heavy metals, drought, high temperature, and salinity and nutrient stress, for developing climate-resilient crops. He was instrumental in engineering non-food crops for phytomeditation of heavy metals and metalloids, and also dealt with the metabolic engineering of oil seed crops for increasing oil content of food for food. Biofuels and value-added bioproduct. He studied the role of glutathione homeostasis of enhanced oxidative stress tolerance in plants. Professor Dhankher has also utilized nanomaterial as a nanofertilizer and nanopesticide for sustainable agriculture and food safety. He has explored the molecular and physiological strategy for developing arsenic-free rice. Professor Dhankher received Outstanding Agricultural Research Scientist Award from the Association of Agricultural Scientists of Indian Origin, that is called SIO in 2021, Haryana, and followed by Saman Award from the Government of Haryana State in India. Many awards, he is a recipient of many awards. I could not read about. Almost 15 awards he is having, Nobel Awards all. And finally I'll end with the Plant Genome and Crop Science and Food and Energy Security. He is a member. He had published 120 research publications in reputed international journals and has 5 international patents. He has achieved a total 6,500 citations and Hutch index of 44. With this, I proudly invite Dr. Dhankher for the second lead lecture on strategy for arsenic phytoremediation and limiting arsenic accumulation in rice. Thank you very much.
Thank you, Dr. Gomti, for the elaborated introduction. We can save time to cut down shorten the introductions. Thank you very much. And also, I would like to take this opportunity to convey my sincere thanks to my friend, my collaborator, Dr. Jose Pathur, and all his students, and also Dr. Shakira, that she hosted me at Kannur University, Dr. Abdul Salam. So thank you very much and I really enjoy this beautiful occasion here, as well as this nice little warm weather. Thank you. Because the time I left from Boston on Friday, you said Sergey had 2 days visit, transit or travel. I had 3 days. And the temperature was -2 at the time on the morning. Anyway, so this talk is slightly different than the theme of the conference on abiotic stresses, and the blame goes to Dr. Jose Pathur. He asked me to give this lecture on arsenic, because yesterday I gave the talk in Kannur University on abiotic stresses. So here he asked me, please talk about arsenic. So I will do my justice, and so what I'm going to do is, I'm going to show you some of those my work and some of those strategies, how I can limit the arsenic in the food chain and also clean the environment. So this is my beautiful campus, University of Massachusetts. Few of the audience has already visited there, and this is the university, this is the tall library and campus pond, and then our state-of-art, this one is not going to work. Our state-of-art facility for life sciences laboratories, we have perfect 4 weathers, a very beautiful scenic valley, and also we have a lot of snow. So, and in the fall, these are not painted. These are original images. I just took it last week on the weekend. This is the perfect fall foliage, the best in the world that we have the fall foliage there. So I invite you to come and visit Massachusetts. Thank you. So what arsenic is doing? Arsenic is an acute toxin. It's a carcinogenic. It causes several forms of cancers, and cancers of liver, skin, kidney, and many of those other health defects, and the blisters and all those. So it's an acute poison basically, and it has been used as the most kind of the #1 poison used to kill people actually in the past. And even Napoleon was supposed to be, there are theory about that Napoleon was poisoned by arsenic. So I didn't, and then arsenic contamination is widespread. So here, most of the sources of arsenic is underground. And underground, because of the soil formation, a lot of these volcanic activities, the arsenic is deposited in the underground rocks, and underground water is contaminated. A lot of activities from the mining and smelting, and also naturally occurrence of soil. But in the past, lead and arsenate has historically been used as herbicide and pesticides to control the disease in the orchards. And also, lead and arsenate were used in cotton field to spray the cotton field to kill the foliage so that they can harvest the cotton balls with the machines. So there is a lot, being this non-degradable metals, arsenic is metalloid, so they stay there forever in the soil and water. And also, farmers are digging up tube wells and extracting the underground water and that's contaminated with the arsenic, and they irrigating the paddy fields, and arsenic built up in the topsoil there. And India has a really very critical situation with the arsenic, especially in the drinking water, and West Bengal state and the eastern part of UP, Bihar, part of Haryana, Punjab. Almost 10% of the Indian population is affected by the arsenic, and maybe more, in the water alone. And you can see mostly the arsenic deposits are mostly in the Gangetic basins, and then in the west, in the eastern state of those. And here the country of gods and goddesses are blessed. We don't have arsenic in Kerala much yet, so maybe not discovered yet. Maybe the blessing by the gods and goddesses, right? So what are the arsenic forms available in the environment? There are two species of arsenic forms: organic form of arsenic and inorganic form of arsenic. Inorganic form of arsenic is mainly arsenate and arsenite, and organic forms are monomethyl arsenic, dimethyl arsenic. Inorganic form of species of arsenic are most toxic. Arsenate and arsenite are more toxic, and out of those two, arsenite is the potent toxin. And how this arsenic in the food system is affecting, especially in rice. So since we started growing the high-yielding rice and wheat varieties after the Green Revolution, and that requires standing water and a lot of fertilizers. So what farmers were doing to meet the irrigation demand, they are digging up tube wells and pumping the underground water from the shallow aquifers that contain arsenic, and they irrigate the field, and water evaporate, and due to repetitive irrigation, a large amount of arsenic deposit in the topsoil, and then when you grow rice is very much adapted to, under flooding conditions, to accumulate arsenite. And that's how this arsenic is coming into the food chain. But you grow these vegetables and other food crops on arsenic-contaminated soil that accumulate that and that affect the food safety. And rice is well designed, it is naturally adapted to accumulate more arsenic. And like here you can see many of those studies shows that arsenic in rice is a very high level globally, in India, in Bangladesh, in many parts of the country. And recently in US, because of the arsenic and cadmium and lead and mercury level was found to be very high in baby food formulas. And the US Congress mandated these two studies, and mostly these the baby food, the companies, the Gerber and all these, they make the baby food formulas is very high level, 10-fold higher than even the parent, like in rice alone. So it's very high level, and because of that, US Congress mandated the Baby Foods Act of 2021, and the demand is closer to zero action, means no arsenic in rice or other food crops, zero arsenic. And that's a challenge for scientists, soil scientists, plant scientists to deliver that. So what are the strategies? How we can limit arsenic in the food chain and how we can clean the environment? So there are several strategies, but there is no single magic bullet that can work. So there are various strategies you can apply, genetic engineering, genome editing approaches to manipulate the genes to exclude the arsenic, prevent the arsenic accumulation. Also there are some agriculture practices that soil amendments with the engineered nanomaterials mineral fertilization, silica, sulfur, and iron kind of fertilization, water management practice and all those. Also, phytoremediation of arsenic-contaminated soil and water. And then, as Dr. Parkash said this morning, that the rice has a very high genetic plasticity, and I'm sure there are wild cultivars that exist in the nature that may have capability to exclude arsenic. So we need to identify those and then introgress those traits through the marker-assisted breeding and develop those arsenic-free rice. So I will show you few of those, what I have done, my lab has done in those. So on that one, how arsenic is getting into rice. So here, arsenic is basically arsenic and silica, arsenite and silica behaves very similarly in the soil, and in rice, rice is well adapted to accumulate very high level of silica. Around 20% silica is in rice, and then these silica transporters, LSI1 and LSI2, here, these are the silica transporters. They transport arsenite from a soil to the root to that, and then translocate all the way to the above ground tissues, and then it goes in the, also it goes in the seeds. But if you manipulate, if you knock down these two genes, LSI1 and LSI2, which are silica transporter, then it will affect the rice yield and rice performance, because silica is absolutely required for rice in a high concentration. And plants could be sensitive to disease and other kind of those yield penalties as well. And so in rice grains, the tomography of this image shows that in the rice, the arsenic is basically in the ovular, this vascular transport system here, in that one. And it's very, if you wash it, some of those will be, before you cook rice, if you wash it for a little bit longer time, some of that can be can be washed off, but it is staying in the traces there, so it's difficult to get rid of that. And if you knock down the LSI1, which is silica transporter, plants become sensitive to insect damage, and also there is a significant yield penalty as well. So we cannot manipulate those silica transporters to just get rid of arsenic. So what are the other alternatives? What we can do? So alternatives is, because this LSI1, LSI2 are the members of aquaporine family. Aquaporine family is a large family. There are subfamilies of aquaporins, PIPs, TIPs, NIPs, and SIPs. And NIPs are basically, the LSI1 is belong to NIP, so we should not be manipulating this, otherwise it will affect the rice performance. So what we did was, we look for those alternative members of the aquaporine families, PIPs, whether they are transporting arsenic transport in rice. And these are those talented people that they work for my lab, and this work was basically started by Kundan Kumar, who is a faculty at BITS Goa campus now, associate professor there, and Kareem, he's a full professor in Dubai, and Sudesh Sikara, and the current postdoc, Ahmed. So they did this work on this project. So here, we cloned some of those. There are 11 members of PIPs in the rice, and we cloned some of those, around 8. And then we did the genopus oocyte, what Sergey said about the electron transport, and in the frog's egg, genopus oocyte, and we use those and to see whether these overexpression of these aquaporins, the PIPs, transport arsenite in the genopus oocyte. And compared to the positive control, which is the LSI1, many of these PIPs also co-transport arsenic as well. So we want to see whether these, manipulating these can have impact in arsenic accumulation in rice. So we heterologously overexpress these in the Arabidopsis, and overexpression of these, like here is the wild type, here is the transgenic lines. Overexpression of these, like one of the member PIP1-3, PIP2-6, they provide strong tolerance to arsenite. But that's the heterologous system. But they have strong tolerance, and there was no accumulation of that arsenic. The accumulation was not differing in the wild type and transgenic. So we were puzzled, why is that? There's a lot of water coming here. That's risky too. maybe, yeah, I'll be shifting on that side, yeah. So they were tolerant. They were tolerant, but there is no accumulation. And we look at that, why there is no accumulation. Then we did the efflux and influx studies, short-term efflux, influx, and we found that in short-term, like 1 hour, 2 hour, 4 hour, compared to wild-type, transgenic lines, they are influxing more in the shoot and as well as in the root. And then after 4 hours, we did the influx for short-term efflux, and then here after 4 hours, here is the transgenic lines, here the wild type, and it's efflux out. So basically, and when effluxing, proportionally, the more arsenic built up in the media. So that shows that these transporters, PIPs, are bidirectional transport. They pump in according to the concentration gradient. When there is more arsenic in the media, then the more arsenic goes in the plant root. And when high arsenic built up in the plant roots, they start pumping it out. They reverse the function. So there are very few bidirectional transport pumps. So our role was to see in planta function of these genes, whether they can have impact on the arsenic accumulation and tolerance. So we use the RNAi approach. I'm not going into more detail about the technology. I'm just going to show you the key results. So basically we make the hairpin loop structure for the RNAi, and then we introduce this gene into rice by the tissue culture methods, and then we get the various transgenic RNA lines, and we see the expression loss of transcript. So you can see there is a significant loss of transcript compared to the wild type in these transgenic lines for the PIP2-6, also for the PIP1-3 as well. So we use 3 of these lines for further analysis. And then to see the accumulation, we designed the experiment in 2 ways: short-term hydroponic uptake and long-term soil studies, pot studies. In the short-term hydroponic uptake, like 8 days, and we just grow the plants in those kind of very low kind of quality technology that we designed this from the pipette tips boxes, and we use those for growing rice. And so we did the hydroponic short-term and the long-term maturity studies. And when the short-term, the results I'm showing you, knockdown of PIP 1-3 and 2-6 both basically showed less arsenic accumulation compared to wild type in the shoot and root in the short-term hydroponics seedling stage. Both genes show the same results. So this shows that these plants are translocating less arsenic from the root to shoot because there is no difference in root. No difference in root, only difference is in the shoot. So that means there is a less translocation from the root to the shoot. The total amount of arsenic may not be affected from the soil to the root, but translocation is affected. And to see that, whether this is a root specific, and we saw that from the previous studies that this PIP1-3 is root specific, specifically localized in the endodermis and this exodermis. And here we also see the transcript abundance in the transgenic lines, in the wild type control plants. It's more of the transcript abundance is higher in roots, and for PIP2-6 is both in shoot and root. So PIP1-3 is more root specific, and we are doing more localization studies in those. But then the key results was we want to see whether there is a less arsenic in the seeds that we eat. So in the long-term studies for the maturity level, we grow the plants in the pot studies, and here again you can see there is a less arsenic in the transgenic RNA lines compared to wild type in the shoot. There is not much significant difference in the root except this in this one, and then there is a less arsenic in the flag leaf and almost 20-25% less arsenic in the grains. So that's in the pot studies. In the field studies could be very different as well. So basically and also we not only knock down by RNA, we also overexpress these, whether the overexpression can increase arsenic accumulation more. So here in this model, when we knock down this RNA, PIPs using the RNA, there is a less arsenic going in the shoot. But when we overexpress these genes, there is a more arsenic going in the shoot and grains. So it's basically reverse. So it shows that clearly shows that these genes, PIP genes, are involved in arsenic translocation or transport in rice. And then second thing was soil amendments. So we use the engineered nanomaterial, mainly the sulfur. Sulfur is the most abundant nutrient that plants require that in high amount, and we use the sulfur instead of regular sulfur. We use the sulfur in the nano form. And here, the results I'm showing, because nanotechnology is growing big, there are a lot of approaches for the nanotechnology in the different fields. It's the fastest growing field in our modern history in any of the sciences in nanotechnology. Every year, there are more than 20,000 publications are there. So, so we use the nanosulfur materials, and this PhD student, Sudhir, and Ahmed was PhD student at the time, now he's postdoc, and Sam is also PhD student, and Jordan master student. They work on this project. So we published this paper showing that nanoscale sulfur improves plant growth as a fertilizer, increase yield, but also significantly decrease arsenic in the grains. So here you can see the pot studies. This is control plants exposed with only with the nanosulfur, plants exposed to arsenite plus nanosulfur, and arsenate plus nanosulfur. You can see plants are bigger here compared to wild type, but arsenite is toxic, arsenate is toxic. When you add nanosulfur, growth was recovered. And when we grow these plants at maturity in the pot studies, you can see there is a significant decrease in arsenic accumulation with the nanosulfur treatments. Here is the arsenite alone, and this is in the shoot, this is in the root, and here is the arsenite plus nanosulfur. There is almost like a 45% decrease, and with the bulk sulfur, regular sulfur, there was decrease, but it was not significant. And arsenate, we just ignore that because rice uptake arsenite, not arsenate directly. And here, but in the root, it was almost 75% decrease in the arsenic accumulation. The nanosulfur amendment with the soil, 75% decrease in the arsenic accumulation. But most important parts are the grains. Here, with the nanosulfur alone, with nanosulfur treatment with the arsenite, there is a 60% decrease in the grain. So there are opportunities to do these kind of various practices. Not all the time biotechnology or molecular biology is the key. This is a easily deployable, acceptable technology in the field without going through any regulations, because sulfur is a nutrient. So this can have a big impact, and in the flag leaf there was no significant difference. So then the third part is the phytoremediation of arsenic. Let me get a sip of water. So phytoremediation of arsenic is my long-term interest. I started my postdoc career when I moved to Georgia. I started on the phytoremediation. Dr. Parkash knows that. He visited my lab when I was postdoc there in Georgia. So, and I designed this phytoremediation of arsenic from the scratch. There is only one plant identified, Teres vittata, Chinese break fern, as a hyperaccumulator, but has a limitation, cannot be grown everywhere. It can be grown only tropical climate. So what I did was, I was developing a genetics-based phytoremediation strategy. I call it a suitcase strategy. You can take this technology and insert those genes in any plant species of any native climatic conditions. Instead of introducing new plant species, you can introduce those in the local varieties. So the idea was, in the aerobic environment, in the soil, not in the rice flooded condition, but aerobic environment, arsenate is more prevalent. So plants can take arsenate is getting into the plants by phosphate transporters, because arsenate and phosphate are structural analogs. It's very similar, and behavior is very similar. So arsenate is getting into the plants through the phosphate transporters system, and once the arsenate is getting into that, our idea was we can translocate arsenate to the above ground tissues, and because arsenate cannot bind, cannot be chelated. Arsenite is highly thiol reactive. It can bind to sulfide groups of any amino acids that contain sulfur. So, and arsenite, once arsenite is bound to those glutathiones or phytochelatins, sulfur-containing peptides, it's no more toxic or less toxic, and that's the only form that can be sequestered or accumulated in the vacuoles, in the leaves. So, our idea was to increase the phosphate transporters, but there are already 9 high-affinity phosphate transporters in plants. We don't need to do much. Arsenate is naturally getting into the plants. Our aim was to reduce arsenate to arsenite in the leaves, and then increase the glutathione content to increase the chelation of those arsenite, and then arsenite chelated with the glutathiones and phytochelatins can be stored into vacuoles. But while we were doing it, we also found out there is a natural adaptation in the plants. Plants try to keep the toxic metals below ground to prevent the above ground tissues from damage. So we found that plants has an endogenous arsenate reductase as well. We need to knock down, otherwise arsenate will not go up. So while we were doing it, we just used 2 genes first. I used this arsenate reductase RC gene from bacteria and gamma ECS from bacteria. So arsenate is basically reducing, this arsenate reductase is reducing less arsenate, toxic arsenate, to more toxic arsenite. And arsenite is toxic, it can kill the plant, but if we simultaneously increase the glutathione level by overexpressing this gamma glutamyl cysteine synthase or phytochelatin synthase genes, it can increase the phytochelatin levels, and it can bind to arsenite, and arsenite bound to that is no more toxic. And that was the paper basically very early on in 2002. I published this there in Georgia, and basically idea was arsenate will get into the roots, and arsenate will become going in the shoot, in the above ground tissues. There we can, by a shoot specific expression of RC, we can convert to arsenite and increase the glutathione content and store in the vacuoles. That was the whole idea. So we put these 2 genes together first. Now you can see, because plants converting arsenate to arsenite, they died. They are sensitive. Plants expressing gamma ECS alone, they are moderately tolerant. But when you combine the expression of these 2 that complement the function of each one, and double transgenic plants become super resistant to arsenic. These plants have 17 times more biomass than the wild type non-transgenic plants. And these transgenic lines, the double transgenic lines, they accumulate 3 times more arsenic than the control or the single transgenic lines. So 17 times more biomass, 3 times more arsenic. Per plant basis, they have 51 times higher arsenic accumulation and tolerance. That's a significant. So, and that's basically very close to a natural hyperaccumulator kind of definition. So basically, when we did this one, then we found this is another strategy here. We need to knock down this gene. Then, but I'm not going to show the data on that one. But we want to translate this strategy into field crops. So idea is for phytoremediation of arsenic or heavy metals, the best practice is do not use food crops. Try to find non-food crops, high biomass, fast-growing crops that you can remediate and you don't use the food crops. People accidentally eat those and worsen the situation. So I found Cranberry abyssinica as a member of mustard family. It is basically inedible, non-food crops and a short life cycle for maximum biomass. For phytoremediation, we need maximum biomass. We don't need maturity. And here, it's low agronomic practice. Also, the best trait for this crop is self-pollinated. It does not cross-pollinate, so no escape of gene flow as well. So here, we put those same strategy. We translate that strategy from Arabidopsis to Cranberry, done by this, now this new graduate student from Kenya. is doing this, taking over from these people returned back to India, and he's doing that one. And here, initially what we found, when we overexpress the gamma ICS gene in the plants are tolerant to compared to wild type. And transgenic plants expressing RC, arsenic reductase, were even sensitive than this one. But when we combine these two genes together, RC and gamma ICS, you can see the difference in the tolerance level on arsenate. This is almost 300 micromolar arsenate, and you can see these plants accumulate has almost threefold, fourfold biomass as compared to the wild type plants, and also they have almost double the amount of arsenic accumulation in the above ground tissues as well. And this plant is very robust growth. It can produce almost 30-40 ton wet biomass, and so ideal for phytoremediation. So now basically what we are doing is we are using the nanomaterials to enhance the arsenic accumulation, translocation from root to shoot, and combining all those 5 genes, stacking into a single plant species so that it can be taken from the soil all the way to the root, to the shoot, and to the vacuole storage. So it will be a challenge, but we are doing this one. And the idea is, particularly in context to India, where we have the arsenic problem in the rice paddy fields, we are growing rice, putting more arsenic contaminated water, arsenic built up in the soil, we harvest rice, then we are planting wheat. That's the wheat-rice cycle in India is more common. And so in between the rice and wheat, if we get 4 or 5 weeks, a month period, we can grow this crop, this transgenic cranberry, to clean the arsenic built up after a few years and then grow other crops. So it's a kind of, can go in a cyclic manner, and we can basically maintain the low level of arsenic in the soil, so prevent the accumulation of those. Okay, so here, what I showed you was that the rice accumulate very high level of arsenic, and these silica transporters are involved in arsenic transport, but manipulating the silica transporter is not ideal because it will be affecting the rice yield and functionality. So we need to find alternate transporters that are involved in arsenic transport, and we found members of PIP, plasma membrane intrinsic protein family, and those are the we showed you that by overexpression and knockdown, they showed that they're involved in arsenic transport and translocation from root to shoot tissues and above ground tissues. So we need to kind of manipulate those to lower the arsenic containing rice and other crops. And also we use the nanosulfur as a soil amendment treatment for arsenic, blocking the arsenic uptake or how the sulfur is behaving is basically from our studies. What it shows in that paper is sulfur is interacting with arsenite and bound to arsenite, and arsenite once bound to sulfur, it makes arsenite sulfide complexes. Arsenite sulfide is non-soluble, precipitate out, and hence less arsenic is available to rice. And then also we are doing this phytoremediation approach to clean the arsenic contaminated soil using non-food high biomass crop species. So with that, this is basically the work is done by the talented postdocs and students. I only just give the advice and guidance, but hard work is done by them, and I give the credit to them for all this hard work. So Ahmed just finished PhD, now joined postdoc. Gurpal, Sudhir, and all these people in the green, they are the current PhD students, and these are some of those visiting faculty and some of those honors undergraduate students, and some of those the recent postdocs. The work I showed you was from Sudesh and Kundan, and also this Kareem as well.